Okay, so in the last lectures, we were talking about ways in which you can regulate gene expression by altering the chromatin structure. And we also mentioned towards the end that you can also alter gene expression by altering the DNA directly. So, and that's by adding a metal group to the DNA. And this process of regulating gene expression is referred to as cytosine methylation. And depending on the type of DNA methylation that's happening, you will have a different effect on gene expression. So we mentioned that CG methylation is the most common form because it's the one that's conserved between both plant and animals. GC methylation in the promoters of genes is associated with silencing of genes or gene repression. If there is GC methylation within the gene body, this has been associated with activation of gene expression. There's other types of gene body methylation which can occur in either the CHG or CHH context, and that's often associated with gene repression, but we're not going to focus on that for this class. This methylation status that actually occur is inherited. Okay, so it's inherited through DNA replication and it's maintained during DNA replication. So you can imagine you have your double-stranded DNA, you have the methyl group on the C's, right, which gets methylated. DNA replication, because of the conservative nature, you will have a parental strand and a newly synthesized strand that will not have that C, the symmetric C methylated, right, in the new daughter strand. However, this is recognized by a methylase within the genome, and they will scan those types of DNA and add a methyl group to it. Okay, so then in the end, both strands will be methylated. And this is a way to maintain this GC methylation throughout cell division and DNA replication. The way we would detect, we have used traditionally to determine whether or not a particular cytosine or region within the genome is methylated, have that methyl group, is by taking advantage of methylation sensitive enzymes. Okay, so we talked in the very beginning about endonucleases that can recognize the same sequence, but will cut at different places within those sequences. And these are referred to as isoschizomers. There are some endonucleases that will recognize the methyl group and unable to cleave that particular site because there's a methyl group added. We saw this when we were looking at site-directed mutagenesis, right, where we had the DPN1 sites, which were methylated sites, right, and the enzymes. So you could use DPN1 to determine whether you had a modified base versus a non-modified base. And it's kind of the same concept. So there's two enzymes in that's commonly used to determine the difference between methylation, C that has been methylated and one that has not been methylated. And that's HAPA2 and MSP1. These two enzymes recognize this CCGG site within DNA. And depending on the context of the methylation, either of these two enzymes will be able to cleave that DNA site. So HEPA2 and MSP1, they can cut unmethylated sites. So CCGG, both of these sites can be cleaved. However, in the case of HEPA2, this enzyme will not cut if the cytosine, the inner cytosine is methylated or the outer cytosine is methylated. Okay, so either this cytosine or the outer cytosine, it doesn't matter, it will not cleave. MSP1 can cut unmethylated site, but it can also cut a site if you have a methylation on this C that's present here, this inner C, okay? So the methylation is only blocked when the outer C is methylated. And you can use these enzymes to cleave, so you can extract your genomic DNA, and then you can add these enzymes, and you can determine whether or not there's a methylation site within your DNA. So in this case, you have your double-stranded DNA. These are the sites. You have methylation occurring, particularly at this inner C location. If you digest with HAPA2, it's not going to be able to digest, right? Because this methylation is going to block that site. So you can get you will get a larger fragment, okay? Compared to if you digest with MSP1, it's not blocked by this CG methylation in this inner site, right? So it can cleave there. So you can cleave this fragment into 3 different pieces, and you can run this in a gel, and you can see, okay, which of your DNA or your regions of your DNA is methylated or not. This was, traditionally used. It might still be used in some labs for directed analysis, but a lot of the work that we do now is looking more on a genome-wide scale, right? So you want to look genome-wide, how methylated is the genome, depending on the organism that you're working on. And we use a method that requires isolating the entire genomic DNA and using a chemical called sodium bisulfide. Sodium bisulfide is a chemical that when you add it to your DNA, this will change your cytosine to uracil, okay? So all of the unmethylated cytosines will be converted to uracil. If it's methylated, there's no conversion, okay? So you can use this method. You can isolate your genomic DNA. So this is genomic DNA. You have some Cs that are unmethylated, some that are methylated. You have a control where you're not going to add any of that chemical to. And you can then add this chemical treatment. And then you can either PCR amplify the product and sequence it, okay? So in your bisulfide treatment, all of the unmethylated Cs will be converted to a U's. When you sequence it, right, because you're sequencing this strand, you will have a T, okay? So you would run this in a gel, in a regular sequencing gel. Of course, it's now automated, and you can read that sequence. You have the comparison of the control. So in your control, you didn't add any of that bisulfide sequencing, so these Cs are not converted, right? They're still Cs. So when you sequence those Cs, what you will end up with is the Gs that correspond to those, okay? So then you can sequence this, these 2 sets of samples, and you can see where there was a methyl group that was added, right? Because this will still be a C that's present here, okay? Compared to if it was here, you would have had a T that was converted. Okay. So this is what we use to measure genome-wide whether or not sequencing, whether or not DNA methylation is actually present within any region within the genome. The way that these methylation marks are maintained, right? I told you that it's inherited, it's maintained from one generation to the next during DNA replication and also cell division. The enzymes that add these methyl groups and also maintain these methyl groups scans the DNA to see if there was a mark that needs to be added, are highly conserved proteins. They're very similar across plants and animals, plants and humans, but their names are different, but they do very similar function, and they have very similar domains within the protein. So MET1 and DNMT1 is the methyltransferase that's mostly responsible for maintaining the methylation of CG sites. And if you look at these 2 proteins, you can see that they're highly similar, right? So the gray and the pink are things that are shared between the 2, okay? So they're highly similar. We also have additional enzymes that are involved in de novo DNA methylation. So if it's going to add a new methyl mark that wasn't there before. So these are referred to as domains rearranged methylases. They're also present in mammalian cells, some conservation with plants, right? So this blue is what's conserved. And then you have these plant-specific metallases, which are the CMTs, which is only found in plants because these are the ones that we know play a role in maintaining the methylation status of CHH and CNG sites, which we, if from the previous lectures I mentioned, we haven't found a prevalence for these type of methylation in humans thus far. And similar to histone modification, DNA methylation can also be spread along the chromosome with the help of histone modifying enzymes, right? So the writers and the readers also can recruit methylases to methylate certain regions, certain cytosines in that DNA. And this is just a graphic showing you that interaction. So as you mentioned before, you have your reader and writer molecules, which, you know, add the mark, read the marks, and keep and start to compact the chromatin, right? So in this case where you have condensation, because it's chromatin condensation, it's associated with gene silencing, right? So you're compacting the chromatin. This reader-writer complex can also recruit DNA methylases, right? So it will recruit these DNA methylases to come in and methylate DNA that's wrapped around these histones, and further compacting it and silencing that region. Okay, so there's an interaction between two, and that can also spread along this DNA. So they work together to achieve a very similar outcome. You want to regulate gene expression. So you're regulating gene expression by altering chromatin structure, modifying the histones, or perhaps you can also do this in terms of methylating the DNA. Okay. So I'm going to just summarize that from the last lecture. We mentioned chromatin, right? Chromatin level of regulation, where these nucleosomes are important, the histone proteins with the DNA wrapped around, including the linker proteins. Whether or not they're compacted or less compacted will determine whether or not you're going to be activating or repressing gene expression. And this is all, you know, happening to help to facilitate transcription, right? So you need to be able to access the DNA, you need to be able to access the promoter to efficiently have your transcript being made. Today's lecture, we're going to talk primarily what happens after your transcript is made, prior to being exported into the cytoplasm to be translated. Okay. So when you're thinking about studying for the exam for lecture 10, in addition to what's on the study guide that's posted, you want to think about questions that, what is the basic unit of chromatin? I think I already mentioned this to you guys previously. And then also think about two of the ways in which you can change the chromatin structure, right? And there's at least two ways that you can do this, and really understanding what are those two ways and what might be the proteins that's involved or in that kind of modification. Okay. So today, so for the rest of the lecture today, I want to talk about more post-transcriptional regulation, what's happening after the mRNA is made. So as I mentioned previously, in prokaryotes, you have transcription and translation that can occur simultaneously, because there's no compartmentalization between the organelles in prokaryotes, right? So as your transcript is made, you can start translating that right away. In eukaryotes, it's a little bit more complicated, as you're aware of, right? You have a nucleus, translation happens in the cytoplasm, so your transcript needs to get from where it's made, where it's synthesized in the nucleus, and then out into the cytoplasm to be translated. Between these two steps, there's many different processing that needs to happen to ensure the integrity of this RNA. Okay, so there's many different processing steps that needs to occur. While in prokaryotes, you can have, we talked about the way that you can terminate. So you have your RNA being made. There's particular sites, termination, transcription termination sites, which usually involve the hairpin structure, right, in prokaryotes. that says this is the end of the transcript that needs to be made, and then that RNA is then translated into proteins, right? Or simultaneously being translated into protein, and that protein ends where the termination site is. In eukaryotes, termination is not well understood. It's a very complicated process. So there's modifications that needs to be made to the RNA, depending on where that transcript ends, before it can be exported and translated in the cytoplasm. 3 key processes that need to occur, that's absolutely crucial for that to happen, is you need to protect the 5' end, and you do this by adding a 5' cap. This addition of the 5' cap also helps to facilitate other aspects of translation later on. And you also need to protect the 3' end, and that's by the addition of a poly A tail. So that's a string of adenines that needs to be added to the 3' end of that RNA. And then you also need to make sure that you're going to be translating the correct transcript. And to do that, you need to remove non-coding information from that transcript. And that's where RNA splicing is important for this process, to happen. So as I mentioned, you have your transcript that's usually made. So after RNA, so when you have your polymerase, you're synthesizing your RNA, what you have is this transcript, okay? This transcript is the information, includes the information for your coding sequence for your proteins. And within many genes have these pieces of DNA that's between the exons that doesn't code for anything, okay? This transcript that's made is often referred to as the pre-messenger RNA or pre-mRNA. And you might see this in literature or in talks where people are talking about pre-mRNA, right? So this is the transcript that's first made. Gets modified by capping, A-tailing, and splicing, right? And this final processed mRNA is usually referred to as the mature RNA, okay? This mature mRNA is what gets exported into the cytoplasm and what gets translated. Addition of the 5' cap is also a very stepwise process. And it's usually occurring as soon as you have about 20 to 30 nucleotides that has been transcribed within that RNA, okay? As I mentioned, the 5' cap helps to protect the RNA from being digested with enzymes that's floating around in the nucleus. So as soon as you have enough of that RNA that's being made, which is 20 to 30 nucleotides, you want to protect that 5' end. And you do that by adding a cap. The steps for this cap addition involve many different proteins. The first is an RNA triphosphate. This RNA triphosphate usually cleaves one of the phosphate groups from this 5' end, right? So it needs to remove it. The reason it needs to be removed is because you have another enzyme, guanylyl transferase, which will actually add this guanine group to that 5' end, okay? So you're going to be adding a GMP, right? So it's G with just one phosphate that will be added to this group, okay? So it comes in as GTP, one of those phosphate gets added. Very strange 5' phosphate addition, but this is the bond that's formed. And then you have a metallase that then comes in and add a metal group to position 7 on this guanine. Okay, so then you have this 5' metal group that's added, this guanine group that's added, and that's what the cap is. This addition of this cap, after this happens, you can also have additional regions in other bases in that messenger RNA that's also methylated. So you can actually also methylate at what we call like a second cap right, to another nucleotide downstream, to the next nucleotide. And this actually happens after this first cap is added. Having multiple caps, 1 to 2 caps, again, really reinforces that protection of that 5' end. And once this occurs, once you have this addition of this 5' cap added to this end, then you will have a protein that gets added, which is a cap binding protein that will get added to this end. So it will recognize this cap binding protein will then be added to this 5' end. This addition of this cap, again, as I mentioned, the importance of it is really to protect that 5' end from being chewed up by any kind of RNAs that's present within the genome, but it also helps to recruit this other protein, this cap binding protein, which is really important for translation because it helps to, it interacts with the ribosomes, as you'll see later, either in this lecture or in lecture 12 when we talk about translation. So these are the steps again: addition of this 5' cap, which is a guanine, which gets methylated. You know that methylation occurs with methylases or methyltransferases, depending on the context. And this is really occurring again when you have 20 to 30 nucleotides that has been synthesized already. As I mentioned, the function of this 5' cap is really to protect the mRNA from degradation. So having that cap helps to mask that end, so those endonucleases, or those endonucleases are not those more exonucleases, because they're also serving at the end, and also the endonuclease that can cleave within the piece of RNA is not able to chew up that or degrade that RNA. It's also important for enhancing translation because the protein that binds to that cap interacts with the ribosome. It's also important for transporting the mRNA into the cytoplasm by transport proteins. And also it's important for proper splicing of the mRNA, okay? And the question is, what happens if the mRNA is not capped in the end? A lot of these mRNAs get degraded, right? So it's single, and that's why mRNAs are so unstable, right? Because they can be easily degraded. So if there's no 5' cap, and you'll see you need other types of protection as well, they get degraded very easily. And they'll never make it out of the nucleus into the cytoplasm to be translated. So the other thing that needs to happen in addition to that 5' cap is the addition of a string of adenines, and we refer to these as a poly-A tail. These poly-A tail that gets added is added after the transcript is made, so it's not something that's encoded in the DNA, right? So there's no string of T's in the DNA, and then you make this string of A's, right? This string of A's get added once that transcript is made. So because termination is kind of random, and we don't really know exactly where, if there's a particular termination site, when that transcript is made, there are sequences within that's encoded within that RNA that's recognized by proteins that's within the cell. Usually these sites are downstream of the last exon that needs to be translated, okay? And these sites are referred to as polyadenylation sites. As you'll see, they have special features. So once these sites are recognized, you have an enzyme that will come in and cut at those sites, okay? Wherever this site gets cleaved, the RNA that gets cleaved off gets degraded. And what happens is your transcript now, that end of that transcript that you cleave, you will add a string of poly-A's to that transcript, okay? And usually they vary depending on the gene or depending on the transcript. So you can have 50 to 250 poly-A's or A's added to the end of your transcript, okay? What determines where that mRNA is going to be cleaved, or where that polyadenylation sites are, We know this from looking at, hundreds of different mRNAs within eukaryotes to look for features that might be similar to indicate whether or not this is the site that's being cleaved. So if you were to look at a group of mRNAs and look at features that's downstream of the last exon for most of these mRNAs, you can see this is what we call a consensus sequence, right? It's consensus because you're having very shared sequences across multiple different transcripts. And you might think, okay, you can probably find this sequence somewhere else in that RNA, right? Not necessarily at the end. What is also important is not just having this particular set of sequences, but you also needs to be followed by about 20 base pairs later by this GU or U-rich sequence. Okay, so the combination of this particular polyadenylation signal and this GU or U-rich sequence is what really adds specificity to where that polyadenylation site is and where you're going to have something cleaving. Depending on, we said, consensus sequences are sequences that are shared. These are the sequences. These numbers represent the occurrence over, so if you're looking at percent efficiency, right? So if you're looking across multiple promoters, 98% of the time, you'll have an A at this position, or 96% of the time you have an A at this position, and sometimes you have a U. Okay, when you have this particular sequence, you have really high efficiency of quantity denylation, and that's what's shown here, right? So this is the core or main consensus sequence. When you have at the second position, maybe a U present, right? So that's like 12% of the time. You also have a high efficiency, but not as high as the consensus, okay? So you can have these two versions, but this is really the consensus that's there. Yes, Christian, sorry, I wanted to come to finish that, yeah.
No, it's okay. You actually answered my question because I was going to ask then the AAU AAA there to the farthest right, then that is the highest efficient then for being read to get the poly A tail. And then that's typically followed by the GU or U rich.
Exactly.
Okay.
Yeah. So this is important because, so what happened, the reason that you have this polyadenylation site, which is this conserved sequence, and then you have this GU or U rich region downstream, the enzymes that recognize and help to cleave that site recognize these sequences. So CPSF, which is cleavage and polyadenylation specific factor, actually binds to this polyadenylation site. When it binds, it recruits additional cleavage factors and one of these cleavage factor actually binds to this GU element. Okay, so it's a combination of having these sites within proximity to each other, that's important, and having something as close to the consensus as possible will determine how efficiently you have these polyadenylation happening. And this is your polymerase. So you can see that this is happening as your polymerase pass down, you've already transcribed all of this region. These enzyme comes in, cleaves at this signal. When it cleaves this site, where you have your 3' OH remaining, you have a poly A polymerase that will come in and add a string of nucleotides. So first you have 10 A's that get added first, and then in the second phase you have a very quick addition of additional A's, and you can have up to 200 additional A's added. And by doing so, you will then be able to recruit this poly A binding protein that also helps to bind to this end to facilitate the next step of this mRNA. So a 5' end, you will have your poly A binding protein that gets recruited because of that cap. At the end of this poly A site, you have this poly A binding protein that gets facilitated here. Yes, sorry, Christian, yeah?
Sorry, I was just curious real quick because is the PAB2 the same as just PAB?
So no, so PAP.
No, PAB, the poly A binding protein, is that the same as the PAB2?
Yes, it's the same thing. It's just, yeah, this is the same thing. Yeah, this is
Okay.
just, yeah, the name of nomenclature is also there. Yeah. So similar to the 5' cap, the function is very similar for the poly A tail, right? It enhances transport of the mRNA to the cytoplasm, enhances translation, protects the mRNA from being degraded from that 3' end. And it also helps to identify RNAs for degradation. So what that means is that some RNAs have different lengths of A's added, right? So you might have a short or a long poly A tail. The shorter the poly A tail you have, the more susceptible you are to RNAs, right? So you can easily be digested. So the length of the poly A is also very important in this protection from being degraded. What determines whether you have a long poly A versus a short poly A really depends on the transcript and also the condition, right? Because maybe you start adding A's and for some reason, you know, the cell recognized that this RNA is not the most stable, either maybe the 5' is a little bit weak or something is wrong with the splicing and the addition of this A tail stops for whatever reason. The mechanism is really not understood. So it's really just all like hypothesis. But what we do know for sure is that RNAs with shorter poly A's are more susceptible to being degraded. Okay. So the next step, one of the other steps that needs to happen, I shouldn't say the next step, one of the other steps that needs to happen is you need to also be able to remove that non-coding information or the introns from your transcript before you export it into the cytoplasm to be translated. And that's usually involves splicing out these introns, okay? The signal for splicing, similar to how you have with your polyadenylation, you have these sequences that's polyadenylation sites. Splicing is also very similar. So they have the proteins that's important for splicing, which are the spliceosomal proteins, need to know where to splice, right? Otherwise, it will just randomly attack your RNA and just start cleaving pieces of DNA, which you don't want. So there are signatures on your mRNA, which are often referred to as splice sites, that tells the proteins that, okay, here is the junction where you need to be cutting and removing this intron. In the transcript, you have these 5' splice sites, and you also have the 3' splice site. Within an intron, you also have, which is important for this splicing to occur, you also have an adenine that's present kind of within that intron. And that's usually followed by a string of pyrimidines, okay? So in addition to these splice sites, you need to have this A that's present within the center, and this A is usually referred to as a branch point. Okay, and it's absolutely important for the splicing machinery as well. At the ends of these sites, if you were to randomly go online right now and find a gene that have introns, what you will observe is that you always have these particular bases that's always common within that intron, right? So at the 5' end, within that intron, you have this, it's usually a G and a U. Because you're looking at DNA, it will be a G and a T, but the RNA will be G and U. And you also have a 3' site where you have an A and G. In the coding sequence, 1 and 2 or 3 and 4, these two positions, they're also important for recognition. They're not as conserved as these two sequences. Quite often, this 1 and 2 is also kind of like a G or a T. But so there are some sequences that you see most common. okay? But within the introns, you have a GU at this 5' splice site and an AG at this 3' splice site. The step in removing that intron from that center includes really the interaction between the phosphate, the 5' phosphate and the 3' hydroxyl group within that intron. So this model is a very well studied and understood model, and it's usually referred to as the lariat model of splicing or the lasso type of model, because when the way that you're removing this intron is very similar to what this formation of this kind of lasso or lariat structure that you have there. Sorry about that. And I'm not even going to try to. Can you see the PDF?
Yeah, we can.
Yes.
Okay, sorry. So I'll just continue with this. I'm not going to try to restart it right now. So yeah, so you have this structure. So what happens is that the 3' hydroxyl from this adenine attacks this 5' splice site region. Okay, so it attacks the phosphate that's here. Because it's attacking the phosphate, right? A phosphate makes a 5' phosphate, 3' OH. They can form a bond, right? The phosphodiester bond. And so it kind of removes, like kind of really evicts, I must say, this intron, okay? So when it removes, when this bridge is formed, right? So this GU with the phosphate and this adenine, you have this OH that's present here, that's left here. This OH will then also attack this phosphate on this 3' site, okay? Attacking that phosphate. Because it attacked this phosphate to form this phosphodiester bond, right? You have a phosphate group here. This gets cleaved out completely. So the intron gets removed completely, okay? And that's what's left here. And this just gets degraded, okay? So for this to happen, you really need to have particular proteins that will recognize, that will help to facilitate this interaction between this branch point or this branch nucleotide and these 5' and 3' sites. And we'll talk about that in the next 2 slides. But the next thing is you want to know how important is it that this A be present, right? That's that A site be present. We know that it's important for proper splicing, right? It is critical. If you remove it, splicing doesn't occur. So if you look at this wild type RNA transcript, you have an intron, you have a proper splice site present within this intron, that A site present in that intron, you will have proper splicing, right? So you will splice exactly at the 5', 3', remove this region. If you were to look at mutants where there's no splice site or no A, that branch point present within this region, you really can't have any proper splicing occurring, okay? Because you don't have that signal, and this signal is really important for splicing to occur. If for some reason you were to have an intron, but instead you move from within this intron, you move this branch point somewhere else, that is where the splicing is going to occur, okay? So you're moving this signal telling it, okay, it's not here, but it's here instead. So it will splice here, which is not correct because you really want to splice at this particular 3' splice site, okay? So proper positioning is also very important where it is relative to the 2 exons. If you move it and put it between this exon, right? And again, you have the branch point here. it will splice where that branch point is because it's not in the correct location, right? And this is, again, you really have aberrant splicing in that context. And let me just put this here. Christian, sorry, yeah.
Yeah, just out of curiosity, because there's so many steps for like regulation and everything. Is there nothing done to ensure that the splice site is in the right spot? Or if something happens to where it's not in the right spot, then the cell actually goes through and terminates that section?
So there's a lot of regulation in terms of splicing that's happening. So sometimes you want to have improper splicing, right? Sometimes, as you will see a little bit later, that's how we create a diversity of proteins, right? So you might have one transcript, and from that one transcript with the multiple introns and exons, you can have maybe 5 or 6 different proteins, right? And that's kind of a regulated process where you really want that to happen because that's the message that you want. When you have a random mutation where it creates some kind of aberrant branch site, like a just natural mutation because something happens and this doesn't occur, many times those mRNAs will either get degraded, it will be recognized that something weird is happening in the cell and it will get degraded, or if it's still goes through the step of being processed, going out into the cytoplasm, then quite often you will end up with a truncated protein. That truncated protein is not functional and that gets degraded. So you can account for these aberrant process that's happening with these splice sites. So it's highly regulated, whether it happens at the mRNA level where you degrade the mRNA, or it happens, maybe it doesn't get exported because you'll see there's also regulation as the export into the cytoplasm, and actually the proteins that are in the spliceosomal protein, some of the spliceosomal proteins are involved in that export process, right? Because they're actually going to code that mRNA and help to get exported out. So there's a lot of regulation that happens to account for this. If for some reason it gets past everything and gets into the cytoplasm to be translated, the protein that's made is usually not functional, and then that protein just quickly gets degraded.
Okay, thank you.
Mm-hmm. That's a great question, yeah. So as I mentioned, so you have these splice sites. How is all of this happening? How is that lariat structure being formed? How is that tack between the 5' and the 3' OH occurring? There are proteins that help to do this, right? So you have this spliceosomal complex, which is made up of proteins and actually small nuclear RNAs. These spliceosomal complex will recognize particular sequences at these splice sites. So U1, we call them SNRPs, or you heard them referred to as SNRPs, right? So these U1 to 6 proteins will recognize these different sites. So U1 recognizes this 5' splice site first. Once it recognizes it, to say, okay, here's where we need to be processing this particular exon. It gets replaced by U6. U2 SNRP is the main player in recognizing that branch point, and that recruits the U2AF, another U2 associated factor that actually recognizes this 3' splice site, right? Because it's kind of next to it. U5 SNRPs bind to both the 5' and 3' site, and they really help to bring those 2 exons after that intron has been removed closer together. Okay, so these proteins really recognize these sites. How do they do that? As I mentioned, these spliceosomal complex have the proteins plus the RNA, right? And within that RNA, there are sequences that share symmetry to regions within that intron. Okay, so U6 comes in, there's that U6 spliceosomal complex, it have this RNA, that small nuclear RNA in there, And that nuclear RNA can base pair with that intronic sequence, like right upstream of that 5' splice site. And that's how it's able to be recruited there and help facilitate that process. That's the same with the branch point, right? So your U2 protein is the one I told you that's mostly involved in recognizing that branch point. And the RNA that's in there will bind. So this is the RNA that's in there will bind to these regions within this sequences or nucleotides upstream and downstream of this branch point and bind to it, okay? And we know that this is important in addition to causing all these mutations. If you were to, by some chance, say you wanted to create a mutation so that U2 spliceosomal complex or SNRPs is not able to directly or to bind as efficiently to the messenger RNA, depending on where that mutation occurs, you will reduce you will inhibit splicing depending on where it occurs, right? So depending on which nucleotide gets cleaved. So here is your branch point. So if you were to mutate this C in this case and add an A, this U2 mRNA is not able to bind as strongly as this, right? So you kind of have this mismatch here. When this happens, you actually have a reduction in splicing, about 50%. However, if this mismatch is just one nucleotide over, so instead of the C, it's the A, you can still, you will still have the association, but you will still inhibit splicing, but you will inhibit splicing by 90%. So you have a greater inhibition, right? It's closer to the branch point, right? So you have greater inhibition. What happens when what can happen when these type of mutations occur is that this U2 SNRP can actually force a mutation within that mRNA so it matches that transcript. Okay, so that splicing can occur. So this is the SNRP, the U2 SNRP, and the mRNA in there. It will force this mutation that's there so that it can actually bind properly to that A site and facilitate it. So it can kind of, what we refer to these as a suppressor mutation. So you have a mutation that's happening and you suppress this mutation by changing your mRNA to deal with that mutation. Okay, so this can also happen in terms of in the regulation of splicing. U5, so that's U6, and we're the U2, which does the branch point. U6 is really, as I mentioned, responsible for bringing the 2 exons together. The RNA in U6 doesn't base pair as well as you saw with the U2 and also U5. But what you have instead is just the last nucleotide of the 5' exon and the first nucleotide of the 3' exon. U5 interact with just those 2 nucleotides and really helps to bridge these 2 together. Okay, and that's really what the job is. So using these different spliceosomal complexes, you're able to cleave the intron and also bring the 2 exons together for proper splicing. Okay. Why, as I mentioned, this Christian asked this question about what happens with the RNA. So we know that RNA splicing is important, right? So many, I would say, many genes in many organisms, eukaryotic organisms, undergo alternative splicing, which is splicing instead of the traditional splicing where all of the exons are spliced out. There's some alterations to that, okay? And this type of splicing it depends on the developmental stage, depends on the gene, depends on the environmental condition, right? So in this case, you have a gene in Drosophila that's depending on whether or not this transcript is this gene needs to be expressed in the female versus the male, the splicing is going to be different, right? So you will have two different versions of this mature mRNA. In the female, you have all of the exons except this third exon, and in the male, you have this third exon present there. When this happens, it creates a premature stop codon. So this protein is functional and this protein is not functional. So depending on the context, you have these alternatively spliced isoform, and this allows you to create many different variations of proteins to regulate different biological processes in the cell. So RNAs that are polyadenylated, spliced, and capped properly, everything looks great. There's nothing to be worried about. Then gets exported into the cytoplasm for translation. They're also not naked, right? So I mentioned that you're having this 5' cap, you have this this cap binding protein, you have the poly A tails, you have the poly A binding proteins. You also have splicing. After the mRNA gets spliced, additional proteins are actually present at those exon-exon boundaries. These are called exon-exon boundary proteins that are present, and these are some of those proteins that you're seeing here. So all of these are present on this mRNA to help it get transported out into the cytoplasm. And once it's out there in the cytoplasm, some of them get recycled to go to the next mRNA, and some of them are remaining because they will help to facilitate translation for the next step. So all of this happens also with the help of RNA polymerase, right? So of course, everything has to be complicated, right? It just cannot be that simple. So you have your transcript, you have RNA polymerase. You guys recall that you have this C-terminal domain of RNA polymerase 2 that gets phosphorylated. And depending on the phosphorylation, or which serine gets phosphorylated is going to be depending on where in the step in transcription is happening. So the capping, polyadenylation, and splicing proteins also interact with this C-terminal domain during transcription, right? So because we observe that you can have the capping protein interacting with this C-terminal domain of this polymerase as the RNA, when you have your 20 to 30 nucleotides to do the cap. And we also have the spliceosomal complex that also interacts with this C-terminal domain. And then you also have the the polyadenylation proteins is also associated with the C-terminal domain. Because of these different associations, we often refer to transcription as being it's happening as the mRNA is being made, right? So it's not really that you have your transcript and then all of these things are happening, right? It kind of happens as the RNA is being made, okay? And then by the time you get to the end with the cleavage, then you can add this poly and it dissociates. So it's kind of almost like a post-transcriptional, co-transcriptional process that's really happening. But there is interaction with these proteins, with this C-terminal domain that's really important for this process to occur. And so when you're thinking about all these different steps in transcription and what happens in eukaryotes, it's really important for you to think about the difference between prokaryotes and eukaryotes, where transcription is occurring in the cell, right? And that's going to be different between these two organisms as well. How and why it needs to be processed, right? We talked about all of the important steps in 5' capping, alternative splicing, and polyadenylation. Understanding the steps in each that's occurring, and also thinking about what kind of experiment you can do to determine whether or not any of these particular proteins might be associating with that RNA, okay? So very similar to when we looked at protein DNA interaction, right? We can do these assays where you're looking, this protein is associating with this DNA, right? We did our DNA footprinting assay. We did chip, we did many different things. Exactly. You can do an RNase footprinting assay, as Hussam mentioned, right? So you want to think about those type of experiments. If I, and very similar to the extra credit exam, if you were to look at a gel, what's happening? If you were to mutate some of these proteins, what would you expect to happen? Okay. So how do we know whether or not our transcript is made, right? So we need to be able to figure out, did transcription occur? Is your gene transcribed? There's many ways you can determine whether or not your transcript has been made, depending on the lab, depending on your question, depending on how much money you have. You can use many different methods, okay? Whenever we talk about gene expression, or is your gene expressed or not, what we're really talking about is how much mRNA is there, how much of the mature transcript is present, okay? And we can measure that, right? So we can look at how much of that mature messenger RNA level is present, and the abundance of that. And that's how we know whether or not the gene is really efficiently expressed or not. That's what we refer to. There's several methods to measure gene expression, but particularly to measure whether transcription even occurs as well. You can do northern blotting of RNA, which is very similar to southern blotting, as we'll mention in a minute. We'll see in a minute. There's other types of assay, run-on assays, reporter assays, reverse transcription PCR, and microarrays and RNA sequencing, which are two larger scale methods, which we'll talk about more towards the end of the class when we're looking more in terms of large scale genomics approaches that are currently being used to study gene expression. So you can look at the RNA levels within your cells. So you can isolate flowers, for example, in a plant. You can isolate the roots or the leaves. You can do the same thing with in eukaryotes, where you just isolate a particular cell type or organ to see whether or not a particular gene is expressed. And you can just look at the levels. Is it high versus low, or is it high versus nothing? Yeah. Northern blotting is very common to southern blotting, right? You guys already are familiar with this. I'll skip over this and go to the next one because it's kind of the same thing. Where you have your RNA, or in this case, a cDNA, right? So the RNA you make, and then you reverse transcribe that and make cDNA. This is representing an expressed gene, right? You can run it in a gel, similar to southern blot, run it in a gel, transfer it to a filter, right? So you have your filter, so you have your apparatus set up, you connect it to a power source, you transfer the information from this gel to this filter. You add this filter into a Ziploc bag, really, and you add a probe, right? So your probes are going to be whatever gene you're interested in. You want to see if that gene is expressed, right? So you load all of your RNA or your cDNA. Then you're going to look for, is my gene expressed? And that's what these little squiggly things look like. These are radioactively labeled probes that represents a particular gene that you're interested in. Okay, so you'll mix them together, they hybridize, expose it to a film, you look at the film and you'll be like, okay, yes, my gene is expressed in this sample, or it's expressed here in this sample, okay? So that's just a general idea, very similar to southern blotting. This allows you to look at different developmental stages, right? Is your gene expressed at different developmental stages? This is an example of a gel, right? All of these wells, 0 to 25, represent different stages in development in frog embryos, right? So depending on when you isolated them, you're going to have different developmental stages. And you want to see whether or not this particular gene, this Nuggin gene, is expressed at these different developmental stages. You see a bright band, and you see in the later stages of development, suggesting that this gene is involved in at that stage in development only for this particular in this particular assay. Okay, and you know that it's only expressed because you're adding the same amount of RNA in each of these lanes, right? And then you're probing with this DNA, this RNA probe, label probe, right, to see whether or not your gene is expressed. The thing about northern blots, first of all, you need a lot of RNA for this assay. You also need to work with radioactivity, so it's not desirable, but this is a technique that people are still using to look at gene expression. When you do these experiments, you need to have a control, right? Because you need to be able to determine, okay, how do you know that you didn't, by some chance, I mean, we're all using pipettes, we're pipetting samples into a well. How do you know you didn't, by accident, added more RNA in this well? And that's maybe why this band is a little bit bigger and a little bit brighter. So you need to have a control, right, for your loading, and also for any kind of changes within that cell. So usually you need to have some kind of what we refer to as a loading control. So a loading control could be, for instance, ribosomal RNA, which is highly abundant. It's made in the cell. It should be expressed in all tissues, right? All stages in development. So you can use ribosomal RNA and probe for ribosomal RNA. So you're loading all of your samples in each of these well. You add looking for your particular gene of interest. In this case, this is just a particular gene, right? NIF-S. And you can see that you're seeing a band saying this gene is expressed. Your loading control, which is your ribosomal RNA, which is always expressed at all times, you can look and see that banding pattern. And you can see that you actually have very similar amounts in these two and maybe these two. You can quantify this, right? I'm telling you that you can see, but you actually quantify this on the computer to know that it's actually the same amount of intensity that you're looking at. And you can see that you have less here. So because you have less here, it could just be that, you just loaded less, right? Because you have less here, okay? Otherwise, all of these will be the same because it should be the same, right? It's ribosomal RNA. You can also use another gene that's called like a housekeeping gene, right? So the housekeeping genes, you guys remember we mentioned these are constitutively expressed. So they're always on there. Depending on the gene, it's not in every cell, but it's always on. Okay, so you can also use, and some people like to use that housekeeping gene as a loading control to see whether or not you loaded equal amounts of your sample in each well. Okay, and in many cases, people use like EF1, which is an elongation factor that's involved in translation because it's an important gene and it's always on because you need to have it around to be able to translate your mRNAs. Okay, so you need to have these loading controls to tell whether or not the difference that you're seeing here is not because you forgot to label, maybe you forgot to load samples here. Okay, so that's kind of what you need to do.
So they're both positive controls.
Both of these are controls, yeah.
Positive controls, if not mistaken.
Yes.
Yeah. Okay.
So that measures, another blot will do mRNA abundance, right? How much mRNA is being made. The mRNA that you see when you look at the northern blot or other types of analysis, such as RT-qPCR, what you're looking at is the steady state level of mRNA. Do you guys know what steady state mean when I say a steady state level of something, steady state level of mRNA? So when you think
fixed rate.
Pardon?
Fixed rate, specific rate, or this?
Yeah. So when you think of steady state, what we're usually referring to is the difference between what's made and what that gets degraded and kind of what's left. So it's what's left of a product after it's been made and after some of it has been degraded. Because as I mentioned, mRNA is going to get degraded, right? Because something is going to happen. Some of it is going to get degraded, right? So you're making it through exactly net production, as Hussam said, right? You're making it, right? And you have this, but as you're making it, some of it is going to get degraded. What we're actually visually seeing, that transcript abundance, is really the steady state level, right? And that's what's there. And northern blots are very good for that type of assays and also other types of techniques. However, if you want to see whether or not your RNA is even made, is it even synthesized, Or if you want to see, is there a difference between how much of that RNA is made, right? You cannot tell this by looking at a northern blot because, again, a northern blot is showing you that steady state level. Okay, so what's been made, what's been degraded, and this is what you have left. It doesn't tell you about how much has been made. So to measure how much has been made, we refer to this as either transcription rate or mostly initiation rate, right? How much RNA has been initiated. And the assay to do that is referred to as a run-on assay or nuclear run-on assay. And that's because you're isolating nuclei. When you isolate the nuclei, what you're isolating is the DNA, the polymerase that's engaged on this DNA, and the RNA that's being made, that's attached to this, that's being read out from this polymerase. So you isolate this, right? Because this is going to tell you, okay, you already started making RNA. And then what you do is, because you're isolating it, there's no more polymerase around, right? So in this pool, if you were to add radioactively labeled nucleotides, if you already started transcription, that means that you will have a polymerase already engaged on this RNA. It will be able to incorporate that labeled nucleotides that you add and continue to extend. Okay, so this is originally what you isolated, and then you're continuing to extend. And this can only happen if you have an RNA polymerase attached to this DNA, right? So that suggests that this is already, this transcription has been initiated and is happening. Similar to what you did with another blot, you would run this on a gel or blot this on a particular membrane, and then look to see whether or not you can detect some kind of expression of your gene. Okay, so these are all the different genes. You can say, okay, in this sample, your gene is present. That means that this has been synthesized and initiated. Okay. This can also measure transcription rate. How quickly is this occurring? And as you can see here, you have this similar, you have this gene X. You add your label nucleotides to it, and you will have transcription happening when you isolate that nuclei, and you can look at it. You can compare that with other genes, right? So you can say, oh, I have these 3 genes, X, Y, and Z. I isolated the nuclei, I added label nucleotides. Let's see how fast they'll continue to initiate, to transcribe their particular gene. Put them on a dot blot, you hybridize them to the probes representing those genes, and you can see, oh, in X, you have really a bright signal. That means you're having really a lot of transcription that's happening, and it's happening very quickly, right? So you can measure rates. This is less, or nothing is transcribed here because it's white. This is maybe a little bit grayish or much weaker than this, right? So you can say that this one is the strongest one. That means that the transcription rate for this gene X is the highest. Okay, so you can measure, again, initiation and also transcription rate. And this is really a comparison, right? When you think of it. So what is the difference between the run-on versus the northern blot when you're studying for this, right? Northern is measuring RNA abundance, right? That's the net level, steady state level that is remaining after it's been transcribed, after some of it has been synthesized, degraded, sorry. Run-on will measure transcription rate or initiation, yeah? So in this particular situation, right, imagine that you're looking at a gene and you're looking to see whether or not these genes have the same rate of transcription or not, right? If you were to do a northern blot, right, so you have 2 genes, number of transcripts over time. If you were to do a northern blot, when you do a northern blot, what you will see is northern blot gives you what is left, right? So this net level. If you do a northern blot, you will say, oh, this gene here probably have much more transcript being made because the net level is much higher. Okay, so maybe you say, okay, this has much higher because you see this band, this net level is going up, right? Compared to this one, which is straight. In order to distinguish between these two assays, you will look at the synthesis as well, right? When you do the synthesis, that run-on assay, this run-on assay is going to give you this part, the synthesis. You see that you have the same rate of synthesis, right? You have the same transcription rate. These are exactly identical. What is different between these two is the degradation, right? So this gene is degraded, this gene is degraded less, and this one is degraded more, right? So you have a higher level of transcript abundance, which you're detecting by that northern blot. Okay, so these two assays really help us to distinguish between, again, the steady state or net level of transcription and also the transcription rate or rate of initiation. Okay, so they're two just very different assays. They all look at gene expression. It just tells you what is happening at that within that level of gene expression. Okay. The other assay that we use to measure gene expression is we can use a reporter. We can use something that tells us this gene is expressed by looking visually, right? Something that's a visual reporter. Here again, your favorite reporter gene, beta-galactosidase, it's used in many, many different contexts, right? So you can use LAC-Z as a reporter. It gives a blue color, as you guys recall. And you can look to see where your gene is expressed in this embryo, because you can look at the embryo under a microscope and see that you have blue color here, okay? And this is, again, the northern blot. This is the same embryo, but it's just one of these stages of the embryo that you see this expression that's happening, okay? The way you use these reporters is that you need to have the promoter of your gene. So what you have here is the promoter of this noggin gene attached to this reporter, right? Because your promoter is going to direct expression, right? That's where your RNA is going to get synthesized, and that's going to help you to express this reporter gene. So you can see which developmental stage, right? Frog embryos are really amazing because you can actually see the different developmental stage very easily. And it also tells you, not only tells you when in developmental time, but it also tells you where, right? So you can see this strip, that's present here in these particular set of cells, okay? So we use that a lot in labs. You can have different types of reporters. You can have fluorescent proteins, you can have luminescent proteins, but it just depends on what you're interested in studying that you will choose which reporter gene you want to use, right? So reporter genes really just easily tell you where and when something is expressed or your gene of interest is expressed. You can also use these reporter genes to determine what is important for your transcription. What is the minimal region within my promoter that's sufficient to drive expression of my gene, right? So this is very useful for us to define a promoter, right? If you guys remember, I told you, you have these promoter elements that's within your core promoter, within your proximal promoter. That is not all of it, right? You need to know, is it within 100 base, is it within 1,000 base pairs? Is it within 2,000 base pairs, right? If you're looking within a cell. So it helps us to define like really what is required or what is sufficient promoter sequence to drive the proper expression of this gene. And you can use promoter-based assays for those type of experiments. So you would take your promoter of your gene, which includes the transcription start site, and you fuse it to a reporter, and you look at gene expression. You can do pieces of these, right? So you can truncate this and do just a fragment. Is it enough to drive expression? You can do a longer fragment. Is it enough to drive expression? So it really helps you to tell, what is the minimal region within your promoter that you need to get your gene expressed in the proper way. And it can also help you to map where is the end of your mRNA. Yeah, where's your the most 5' end of your mRNA, because you can see where your actual 5' untranslated regions in that mRNA. So it's very useful for that way. So can you clone mRNA? Can you clone RNA? No, because it's single-stranded, right? Whatever you're cloning it into is a double-stranded molecule. Your vectors are double-stranded, right? Or your plasmids are double-stranded. RNA is single-stranded. You cannot clone RNA, so you need to make it into DNA. We talked about this previously, right? So you will use reverse transcriptase to convert your messenger RNA into complementary DNA, okay? Doing this assay, right, we already, reverse transcriptase from retroviruses, right? Using these reverse transcriptase and converting your mRNA into cDNA is absolutely critical for studying gene expression in eukaryotes. One of the reasons is that to bacteria, there's no splicing occurring, right, in bacteria. So if you wanted to express a eukaryotic gene in bacteria, you need to get rid of the introns, okay? And the way you get rid of the introns is by making this cDNA, right? Because you're gonna have your messenger RNA that's been properly spliced and capped and polyadenylated, and then you're gonna make the cDNA from that mature processed mRNA. And then you can express it in bacteria, okay? You can also use this to determine where in a genome you have introns, right? Because you can compare your cDNA where there's no introns with genomic DNA which have introns. So it can tell you precisely where your introns are located within the genome. And it's also used to measure the level of gene expression when you are using other techniques, right? We talked about Northern blot or nuclear run-on where you're directly using RNA in most cases. But you can also use the cDNA to do reverse transcriptive PCR-based method to looking at gene expression. And you can also use it to do microarray analysis, which is also a large-scale expression analysis. Okay. I really wanted to get through the making of the steps of making the cDNA for the exam, but we're probably not going to be able to do that. I'm going to stop right here. Maybe I will give you an extra credit question on the exam related to the uses of cDNA and what it's used for, just as an extra credit because we haven't gone through it, and we'll just go through it in lecture 12 and it will be on the final exam. But so look through it and let me know if you have any specific questions during office hours or before the exam, but it's not required. You don't need to know this, it's not going to be on the exam. I can just throw it in if I wanted to as a extra credit question. So we'll stop at this and I'll post the last slide so you will have the material we covered up to today to study for the last exam, for the next exam, sorry. What I just kind of want to, again, I want to summarize understanding the methods, right? Use the difference between the steady state level versus not, and what is kind of some of the difference between genomic DNA and cDNA. We talked about that, right? One have introns, the other ones don't, yeah? So at the end here, you also want to be thinking about some questions that you might see, right? So I'll also include this, which of the following deal with RNA splicing is true, right? I mean, go through these different steps to see which of these could possibly be true. Again, because of time, I don't have time to go through all of this right now. And also think about the type of gene expression, what type of gene expression is being measured when you're looking at the Northern blot versus a nuclear run-on, right? Super important that you understand that. The exam is going to be very similar to exam 1. It's also going to be very similar to the final exam, right? Where you have 3 parts. Multiple choice, short answers, which include fill in the blank or true, false, or just like one answer you need to. And then you're going to have a few questions, which are more free response. The material is going to cover lecture 7 through lecture 11 to the point where we stopped today. And this is really just a guide to help you to study for this. And then again, if you have questions, let either Israel or I know. If you can make the office hours, you can attend office hours. Otherwise, just shoot us an email. Absolutely important. I mean, this point in the class, a lot of these steps you might think, oh, it's mostly memorization, but you really want to understand the concepts that's there. So if you're asked a question in terms of some kind of experimental design, you can really understand what that is. Okay. And then you will definitely, again, just go through these lectures. Again, key points which I think are important for you guys to know, I noted here. But this is really just use this as a guide to really study, again, to study whichever lecture that you're interested in. Go through them, see which of these lectures might be a little bit more challenging. Let us know. So if you have particular questions about any of these. But this is really, I mean, I don't want to keep you guys much longer, but this is really kind of what you want to go through for studying for this exam. And this is really the last step, which I had hoped to get through. So really not, probably not going to particularly ask about the basic and important steps in making a CDNA, since we didn't get to it again. Could be an extra credit question. If you feel comfortable with it, you answer it. If not, you don't. But that's kind of where we're going to stop. Any questions?
Yes, Professor, will there be backtracking for the multiple choice?
The multiple choice will be backtracking, but you will still have single questions being asked. Yeah, just one question at a time.
Okay, sounds good. Thank you. And also I have a question. Can you give us like an estimate of how many questions per like A, B, or C, just for like time frame wise, just to plan out?
I do not know at the moment. I haven't finished the exam, obviously, so I don't know. I mean, it's going to be a little bit less than what you had before. And again, with that said, no, both the first exam and this exam are exactly in terms of the number of questions as I've done in previous exam, but I do recognize that if you're doing this online, it just takes a little bit longer than usual. So you will have a little bit less questions than I normally do for these exams, just because I'm taking this into account. But I don't really know right now how many questions I'm going to have, because I honestly have not finished preparing all of the questions yet. But it will be a little bit less than exam one.
Okay, thank you.
Jimmy?
Hi, yes, Professor. I just have a quick question. Do you think it's possible or take in consideration of opening all 3 parts so then we can decide
Yes.
which part to work?
Yep.
Okay, thank you.
Jimmy? Oh, Jimmy. Oh, you still have plates. I think Shayla, but I guess I probably answered the question.
Yeah, that I had the exact same question, if they were all going to be viewable at the same time.
Yeah, if they were all available at the same time, then we can open them all together, and then we can backtrack to each one without having to worry about submitting them like singly, and then we just think we have to go back. And well, I'm just saying, if they're all available together, all 3 sections of the test, then that would be most helpful.
Is there a way that we might be able to have it to where, like, there's the part A, B, and C, but they're all together in just one, so that way we can go through the entire thing instead of having to look at individual sections? Or
No, no, that's not. I mean, there's a number of reasons that I don't do that. One of the reasons, every time when I've tried that, the last time I tried this, the iLearn was not happy with that situation, because I have images a lot in my exam. So I'm not even going to try that, because then everything is going to crash, and then we're gonna have a problem. So that's not gonna. Yeah, it's gonna be 3, and I mean, you can just go between them as you're able to. Yeah.
Professor, how can we go between them though? If we complete one, then we have to go to the other, and we can't go back to the other one.
No,
Yes,
she
you
said
can.
that they're
Yes,
gonna
you
be
can.
open.
Yes, you can. So they will all be available. You can work on them whenever, again, the exam is the class period. So when the class start, when the class end, you decide how you want to do them. Yeah, but they're all available. You can work, you can, the only way you're not going to be able to go back is if I tell you that you need to do it in one sitting, which I'm not going to do, right? So you were able to do this.
Understood, Professor. Thank you.
Professor, for the timers, are they gonna be displayed? Are they gonna be?
No, because the thing, so with the timers, as I mentioned, the exam is going to be when the class begin, when the class end, unless you know some of you have exceptions, so you have a little bit of longer time. The exam is going to be when the class begin, when the class end. I am willing to tell you when you have 20 minutes or 30 minutes left. There's no timer because obviously this was an issue the last time, and I don't know why. The problem is when you start an exam, if there's a timer, when you start that exam, so let's say you start your exam, let's say you start part 3 at 12:30. If I give the entire class period for that exam, when you start at 12:30, right, you're going to have the entire class period. But then if you jump to part A, which is also that entire class period, and you jump to part A at 1:30, you're going to start and you're going to see, oh, you have an hour and 20 minutes, but you actually don't, right? So that seems to be what I think a lot of you guys had these issues with, which I don't understand why, because again, the exam is just a class period. The timer is just set there for you to know that, okay, this is how long it should take you to take this exam, right? And it just changes as you start. So there's not going to be any timer because I think that that's going to be, again, confusing again, even though.
Yeah, it was super confusing last time. So yeah,
Right, so it's going to be, so there's no timer. This is your time. You know that your exams, you know when the exam is supposed to start and you know when the exam is supposed to end. So you're responsible to submit this exam at the end of that period. Yeah.
Okay, so no auto submissions then either, right?
So the auto submission is going to happen at the end of the class, right? So when that class ends, right? That's when the exam ends. Right? Like I mean, like every other exam, right? I mean, it's the end of the class. It doesn't really go beyond that time.
No, I understand that. I'm just asking because I've had other classes where we've had multiple sections like A, B, and C, but each section was designated its own time, and that entire time for the exam added up to the total class time. So that's that way, like.
Yes, the problem with that as well, right, which again, I'm pretty sure people would also have an issue with, right? The problem with that as well is that I don't want to say how long you should take on each part, right? So that is why it's a little bit difficult to split the time and say, okay, 30 minutes for part A, 30 minutes for part B, because maybe some people take longer on each different part, right? On each of the parts. It just depends on you and what you feel comfortable doing. So I think there's not going to be any timer. All of them are going to be available at 1:50 when the class ends. Whether you submit it or not, it will be automatically submitted because that's the end of the exam. Okay? That's clear?
Professor, I have a quick question about the lecture.
Yes.
Does the reporter gene technique help with measuring the rate or abundance of mRNA?
It's mostly abundance.
The abundance, okay.
Yes, it's mostly
So there is
abundance.
like no start point like in the run-on essay. That's why.
Pardon?
There is like no start point, right? You just insert the gene and then it start producing like at any time, right? Because like in run-on essays, you have start point where when you add the labeled nucleotide, right?
Yeah, exactly. So you'll just
in gene
yeah.
reporter, you don't have this, so that's why it's abundance, not the rate.
Yeah. Yeah.
Oh,
yeah.
Just making sure. Thank you.
Yeah, that's a good question. Yeah, very good question. Yeah.